as well. Okay. Okay, good afternoon, everyone. Sorry, I couldn't conduct a session in the morning. I had some urgent work of college, which I had to finish. So, anyways, we are here for the second lecture and second session. So, we are going to talk about restriction enzymes today. I'm not going according to the syllabus flow. In syllabus, we were supposed to talk about the gene manipulation part, but before that, I think the basics of restriction enzyme and what do you mean by probes, etc. I wanted to make that clear. So we'll learn about restriction enzyme and then how these restriction enzymes and probes can be used for gene manipulation would make more sense. Okay. So we are going to start with restriction enzymes today. Let me put this on the slideshow. So what are restriction enzyme introduction, and these are the contents we will be talking about, maybe 3 or 4 contents we can finish today. Okay. So in introduction, yes. What do you mean by restriction enzyme? So restriction endonuclease. Yes. Yesterday also I told you about the same. Endonuclease is what? Is the one which, just a minute, just mute yourself.
I'm in middle of meeting. Shall I call? Could you call me back?
So restriction endonucleases are the enzymes which cut the DNA.
At least
are the enzymes that produce internal cuts called cleavages in the DNA molecule.
After 3, not after 2.
So restriction endonuclease, that is restriction enzyme, is a bacterial enzyme that cuts double-stranded DNA into fragments after recognizing the specific nucleotide sequence known as recognition or restriction site. So this is very important. So restriction enzymes, first of all, this definition tells you that are derived from bacteria. They are of bacterial origin. And secondly, they can cut at specific sites, and those sites are known as restriction site or the recognition site. So it depends on from which bacteria you have derived that enzyme and which site it can recognize. So you can modify it, but that would be like, that would be a modified advanced technique that you'll have to do in the lab by manipulating the restriction enzyme. But if you are deriving a restriction enzyme from a bacteria and you are purely taking that restriction enzyme, then it all depends on that enzyme, that which site it can recognize and where it will make a cut in a double-stranded DNA. So restriction enzymes are believed to be evolved by bacteria to resist viral attack. So how these restriction enzymes, why do bacteria produce this restriction enzyme is to kill the bacteria, viruses that can infect the bacteria. Okay, so that is how, that is their process of handling the viruses. So how our body handles every infection through immune response is the same way bacteria handle the viruses, through making these restriction enzymes which will cut the DNA or RNA of the viruses. Okay, and helping them to not to get infected by those viruses. So restriction enzymes are also known as molecular scissors. Very important. So restriction enzymes are known as molecular scissors in the sense they will be recognizing a particular site on the DNA and they will be making the cut. Okay, so if they are recognizing particular sites on the viruses who are going to attack on them or infect them, they will recognize those particular sites, those particular sequences, and cut the DNA or the RNA, and that is how they will break the structure of the RNA, viruses nucleic acids, and will help them to not to get attacked by the viruses. So these restriction enzymes humans have used for many purposes, in the sense to manipulate a DNA and to make sure, like, you can incorporate the copy of the gene that you want to insert in a particular DNA. So you can make use of this restriction enzymes. You can make the cuts into the DNA and insert the DNA of your choice. So that is what the main use of restriction enzymes has been done nowadays. Yesterday we were talking about the plasmid and the vector, right? So plasmid, vector, all this, whenever we are making a cut in a DNA, we most of the time end up using restriction enzymes. So important things that you should know is restriction enzymes are called as molecular scissors. They can cut the DNA at the particular site known as the recognition or restriction site, and restriction enzymes are evolved from bacteria, means they are of bacterial origin. So moving on, um, discovery of restriction. So the term restriction enzyme originated from studies of phage lambda. In 1960, it was shown by Werner Arber and Matthew Messelson, but they studied, just a minute, they studied only about type 1 restriction enzyme in 1970. So there are various types of restriction enzyme, type 1 to type 4, I guess. So we'll look into that also as we proceed further. So in 1970, Hamilton O. Smith and Thomas Kelly and Kent Wilcox isolated and characterized the first type 2 restriction enzyme, which is known as HIND2, and from bacteria Haemophilus influenza. Okay. So for their work in the discovery and characterization of restriction enzyme, the 1978 Nobel Prize for Physiology or Medicine was awarded for Werner Arber, Daniel Nathans, and Hamilton O. Smith. So they were the first one who got the new Nobel Prize, and they recognized and characterized this particular restriction enzyme, which they derived from Haemophilus influenza bacteria, and they named it as HEND2. Okay. Next is types of restriction enzymes. So there are three types that are listed over here, and now we have type 4 as well. So if they, we will talk about the type 4 later on, maybe I'll have to gather the information for the same. So types of restriction enzymes. So restriction enzymes are categorized into three general groups: type 1, type 2, and type 3. Okay. And characterization of restriction enzyme on the basis of their composition, enzyme or cofactor that they require, okay, the nature of their target sequence, what exact sequence of DNA, that means restriction site, कौन सी restriction site वो target करते हैं या फिर कौन सी recognition site वो target करते हैं, on that, and position of their DNA cleavage site relative to the target sequence. So on this basis, we generally characterize the restriction enzymes. Okay? First of all, what is their composition? What enzyme cofactor they require? What is the nature of their target sequence, and position of their DNA cleavage site relative to the target sequence. Okay? Then how restriction endonucleases work? So by far, let us see. We will be seeing the each type into detail. Okay, the type 1, type 2, type 3, and also the nomenclature of the restriction enzyme. So by far, we have learned the definition of restriction enzyme. What all different types of types, means categories, are there for restriction enzymes, and उनकी कैटेगरी कौन से बेसिस पे बनाई है, that is what we have seen. And then how restriction endonucleases work. So restriction enzymes recognize a specific sequence of nucleotides and produce a double-stranded cut in the DNA. These cuts are of two types. Either they produce the blunt end or the sticky ends. Blunt end in the sense they will straight away make a cut, like you're cutting a vegetable directly straight, onion. And in sticky ends, they will be cutting in a specific manner, where there will be some sequences which will be overhanging. Yesterday also I told you about the same. So sticky ends, so they produce some sticky ends, which will be, means that they will be producing some nucleotides which will be hanging in there, and over here there will be a blunt cut, which will not be having any overhanging nucleotides. So in these two ways, the restriction endonuclease enzymes can work. First, it can produce a blunt end, or it can produce a sticky end. And that completely depends on what kind of a restriction enzyme you're using. So it's very important to understand this, that restriction enzymes recognizes a specific sequence of the nucleotide and produce a double-stranded cut in the DNA. And these cuts are of two types. So either a restriction enzyme can produce a blunt end, or it can cut the DNA to give the sticky ends. Okay. Then blunt end and sticky ends. So these blunt-ended fragments can be joined to any other DNA fragment with the blunt ends. ठीक है, जिसके just like you have one blunt-ended DNA and another blunt-ended DNA, with the help of exonuclease, you can join those DNA again. Now make sure what I'm saying right here is exonuclease. Endonuclease is the one which cuts the DNA. Exonuclease is the one which joins the DNA. So both of them are enzymes. So endonuclease, restriction endonuclease are the enzymes which cut the DNA, and exonuclease are the one which can join the DNA. So these blunt ends, these enzymes are useful for certain types of DNA cloning experiments. Okay? So blunt end produced by restriction enzyme are useful when we are doing the cloning of the DNA. Cloning is nothing but you will be putting the gene of interest or DNA of interest into another DNA and making sure that it is producing or doubling the copy of that DNA that you have inserted into another DNA. Okay? So sticky end is DNA fragments with complementary sticky ends. Over here you can see the overhanging nucleotides. So DNA fragments with complementary sticky ends can be combined to create a molecule which allow the creation and manipulation of DNA sequences from different resources. So either you have this kind blunt cuts or you have a sticky cuts, okay, sticky ends. So both of them can be used for manipulating a DNA sequences. So main discovery of restriction enzyme was a very advantageous thing to manipulate the genes. Okay, so ultimately its application went into the manipulation of the genes, where we can insert the DNA of interest into another DNA, where the copies of that interested DNA can be made faster. Okay, for example, we now can produce the insulin. Before the insulin that we used to get in the market, they were horse derived, or if they were derived from different animals, pig derived or something like that. But now we can make the insulin inside the lab. How we have managed to do that? By putting the DNA which can produce the insulin into the plasmid DNA by through this cloning technique. And for this cloning technique, we use the restriction enzymes to make the specific cuts into the DNA so that we get that desired DNA which we can go and stick into the plasmid DNA. And we all know that bacteria multiply at a very faster rate. And this plasmid DNA, if you are able to manipulate that plasmid DNA, it will be multiplying at a very faster rate inside the bacteria. And we will be able to generate lots and lots of insulin enzyme. Okay, and then we can extract that insulin from the bacteria, right? So that is how one of the applications of this where restriction enzymes are used and where gene manipulation has happened has helped us to achieve a very bigger goal. Okay, because before that it was really difficult to use a horse या फिर pig originated insulin, which was very difficult to extract as well. Okay, then yeah, so over here they have shown like how the sticky ends are produced. So EcoR1 is the enzyme which will cut the DNA over here. So it will eat this part, it will eat this part, and it will make the flanking regions. The overhanging regions are also called flanking regions or the sticky ends. Okay, so the DNA fragments joins at the sticky ends. So you can join this sticky ends again, and we can make a recombinant DNA. Okay, so you are understanding restriction enzymes are used to cut simple part of the DNA, where we'll be getting the flanking regions over here also. So these two are the different DNAs, red and blue. Okay, and where we are getting the flanking regions, and later you can join these flanking regions to make a recombinant DNA. That is how the simple experiment works. It looks very simple over here, but it needs lots and lots of caliber and hard work and a very micro observation and lots of technique, right technique to get this done very accurately. Okay. Then moving on, type 1 restriction endonuclease. We'll be discussing about the types of restriction enzymes now. So, type 1 restriction enzymes are complex endonucleases and have recognition sequence of 15 base pairs. So they can basically recognize a sequence of 15 base pairs. Are we clear till here, Supriya? Are we fine?
Yes, ma'am, we are clear here.
Okay.
Yes, ma'am.
Okay. Uh, So we'll discuss about the type then. So type 1 restriction enzymes are complex endonucleases and have recognition site or sequence of 15 base pair. They cleave the DNA about 100 base pair away from the 5' end. So if you have, let me show you. So if you have DNA like this, okay, so it has lots and lots of nucleotides. So this restriction enzyme can detect a sequence which will be 100 base pairs, that is 100 nucleotides away from the 5' end of the DNA. This is the 5' end. This will be the 3',
Ma'am, it's 1,000.
right? And this will be 3'. Somebody's mic is unmute. Mute. Please mute yourself. And this will be
Ma'am, I want to say that it is 1,000 base pairs away from the foundation.
okay, yeah, 1,000 is there. Thank you. So yeah, 1,000 base pairs. So it will be detecting a sequence only after the 1,000 base pair away from the 5' end of the sequence. That is TCA located within the recognition site. So it can particularly detect this TCA part. Okay, so TCA is nothing but the nucleotides, ATGs located within the recognition site. For example, ECO, K12, ECO, B. So these are the names of the restriction enzymes who can detect the particular restriction site and this particular sequence 1,000 base pair away from the 5'. Okay, so the type 1 restriction enzyme work with the help of many cofactors factor like as S-adenosyl methionine hydrosylated adenosine triphosphate and magnesium ion. So basically, these enzymes, if you want these enzymes to work, you need cofactors. Okay, and these cofactors could be SAM, which it could be HAT, and ATP or add magnesium. Means this whole is one molecule only. This whole together is the one molecule only. So basically, for them to work nicely, you need the cofactors. So every enzyme need a cofactor for them to work properly. So these are the cofactors which are needed by type 1 restriction enzymes. And the type 1 restriction enzyme methylates the DNA sequence at the site of recognition. So basically it methylates, okay? And then that is how it cuts the DNA, okay? Demethylation and methylation is very important. So the carbon, we all know the nucleotide structure. It has a phosphate group. It has the purine pyrimidine structure. So one of the carbon which will be methylated, by this restriction enzyme and will be making the cut accordingly. Okay, so for you to understand this part, you have to know the structure of the DNA. You have to know the structure of, like, you know, the chemical structure of the DNA, how nucleotides are, like what all bases are there. Because when I'm talking about methylates the DNA sequence, that means जो purines में से या फिर pyrimidines में से जो एक आधा कार्बन रहेगा उसको वो on one of the carbons they must be adding a methyl group, and that is how they cut the DNA. Okay, the type 1 restriction enzyme translocates the DNA sequence. So it will be translocating the DNA sequence and will be making the cut into the DNA accordingly. Then subunits of type 1 restriction enzymes. Type 1 restriction enzyme possesses three subunits. Is HSDR is required for restriction, HSDM, which is necessary for adding methyl group to host DNA, that is methyl transferase, and HSDS, which is important for the specificity of the cut site recognition in addition to its methyl transferase activity. So these subunits are very important for type 1 restriction to have the cut, to get the DNA cut into two halves, like however they are going to cut, like in the sense whenever they are going to recognize this particular recognition site, and then they'll be making a cut. So HSDR is required for restriction, HSDM is required for the methyl addition of the methyl group, HSDS is required for the specificity of the cut site recognition in addition to its methyl transferase activity. So these are the subunits of the type 1 restriction endonucleases. So these subunits need to be there with the restriction enzymes in order to make sure that the enzyme works. So these are like, members of that endonucleases, type 1 restriction endonucleases, without which the action, the goal we are trying to achieve will not be achieved. Okay. So same is over here, type 2 restriction enzyme endonucleases. So the type 2 restriction enzymes are remarkably stable, induce cleavages either in most cases within or outside their recognition sequences, which are symmetrical. Okay, so they will be ideally making a cut at a palindromic sequences or something like that. What it is, we will talk about it later. So the type 1 restriction enzyme work with the help of only single cofactor like magnesium. In type 1, we saw that it needs 2-3 cofactors to work properly. But over here, they need only magnesium ion. Type 2 restriction endonucleases need only magnesium ion to work properly. The first type 2 enzyme to be isolated was HIM2 in 1970, for which they also received a Nobel Prize. We have already talked about it. So only type 2 restriction endonucleases are used for restriction mapping and gene cloning in the view of their cleavage only at the specific site. So these are the enzymes which are used very frequently when you want to do the restriction mapping and gene cloning. Okay. Okay. And just a minute, let me see if they are going to talk about the palindromic sequences. Okay. So if you see over here, this is the restriction site which is recognized by the type 2 restriction enzymes, and if you see, these are the like kind of a mirror image of each other. If you put a AATT and if you put a mirror in front of them, then you'll be seeing the opposite of it. So those kind of sequences will be called as, which are mirror images of each other, will be called as palindromic sequences. So generally type 2 restriction enzymes recognizes the palindromic sequences and make the cut accordingly. And these are the and those kind of sequences are very are present very frequently on the DNA, and that is the reason these kind of enzymes are used very frequently in order to do the restriction mapping or gene cloning. Then cut of type 2 restriction enzymes. So type 2 restriction enzymes can generate two different types of cuts depending on whether they cut both the strands at the center of the recognition sequence. So the former cut will generate the blunt ends with no nucleotide overhangs. We already know what do you mean by nucleotide overhang. The later generates the sticky or the cohesive end. So they can make two different types of cuts. Either they can make the blunt cuts or they can make the sticky or cohesive ends. So the former cut will generate the blunt ends with no overhangs, whereas the later can generate the sticky or cohesive ends. So a restriction enzyme not necessarily will produce only blunt ends or only sticky ends. It can produce both the ways. It can produce both the kind of cuts in simultaneously in a reaction. Okay. So पहले अगर उसने blunt cuts किए हैं तो उसके बाद it can also make a sticky cut. Okay, and it can means sticky cuts, I mean to say it can cut the DNA which will make the which will have a sticky ends. Okay, so subunits of type 2 restriction enzyme. So type 2, these are the subgroups are defined using the later suffix. So type 2 B restriction enzymes, type 2 E restriction. So these are the subunits in the sense you these are the further divisions of type 2 restriction enzymes which are a little bit different from each other. Okay, so these are like types type 2 restriction enzymes के अंदर वाले types है ये. So they might be having specific differences in between themselves which according to which we can pick up a restriction enzyme, the type 2 restriction enzyme. Okay, so maybe type 2 restriction, type 2b restriction enzyme cuts the DNA in a blunt way. Type 2 restriction enzyme, which is E subunit, can give you the sticky ends. It can recognize a different kind of a sequence or something like that. So something will be common in between them, but they will have a little bit of a difference depending on which where exactly they are cutting and what kind of a sticky ends or what kind of a cut they are making. Either they are giving you a blunt end or they are giving you a sticky end. So next is type 3 restriction endonucleases. So type 3 restriction enzymes are intermediate between type 1 and type 2. So they cleave the DNA in the intermediate vicinity of their recognition site. For example, EcoP1, ECO15, HIN3 are the examples of type 3 restriction endonuclease. They cut the DNA up to 20 to 30 base pairs away from the recognition site. So uh, if this is the recognition site, okay, on a DNA, if this is the recognition site, इसके आगे 20 to 50 base pairs जाके it will cut the DNA. Okay, so they will cut the DNA 20 to 30 base pairs away from the recognition site. This enzyme contain more than one subunit. It can either have it requires ATP and it also requires some magnesium ions. They require ADOMET that we adenosyl methyl transferase and ATP cofactors for their roles in the DNA methylation and restriction. So basically, if you think about it, all the restriction enzymes, they do methylate the DNA and proceed with their action of restriction and then cutting the DNA. Okay. Now the technology has developed in such a way that people are trying to manipulate these restriction enzymes. That means restriction enzymes has three main functions to be done. First is recognition, then restriction, and then cutting. So you want to modify this restriction enzyme in such a way that it can recognize the DNA the way you want it to recognize. Currently over here when we are talking, we just know that type 1 restriction enzymes detects some particular sequence. If it detects that particular sequence, then only it will make the cut. So what humans wants to do is they want to modify this restriction enzyme in such a way that they can cut the DNA anyhow, anywhere as they want, and that is what the CRISPR-Cas9 is doing. The CRISPR-Cas9 is the most advanced technique currently, where we are able to manipulate the gene according to the way we want, and we are able to cut the DNA wherever we want. So if we are able to achieve that, like they are using this little bit of this technology where your restriction enzyme is able to recognize the DNA where you want it to recognize. So it's not up to the restriction enzyme, it's up to you. Like it's up to us where you will want those DNA to, where you want that DNA to be cut down, and how you can insert the gene of your gene of interest into the DNA, right? So if that happens, then it will be very easy, right? Because you can easily then remove the mutated DNA and replace them with the proper DNA, and that way we can take care of the all genetic diseases that are happening, where which are happening due to mutations into the DNA or on chromosomes, if you want to say. And if we cut down that part and replace it with the normal DNA part, normal nucleotides, it will really have a like edge or a breakthrough into gene manipulation. So that is what people are trying to do. Currently, we know that we have restriction enzymes, and those restriction enzymes can cut the DNA at particular site. But if you make a restriction enzyme which will cut at a particular site according to your will and wish, it will be very easy, right? Let me clear this. So it also needs some subunits or cofactors to work properly and conduct the DNA methylation, and further go ahead and have the restriction done, and cut off the DNA will be happening. So nomenclature of the restriction enzyme. So there are no subunits of the DNA, the type 3 restriction endonucleases, I guess. For type 2, we had this subunits BEMT, and then for type 1 also we had subunits, let us see, which are HSDR, HSDM, and HSDS. Okay. Then moving on, how exactly you name a particular restriction enzyme. So nomenclature of restriction enzyme after bacteria which produces them. So EcoR1. Let us look at the EcoR1, how it is named. Genus is Escherichia, species is coli, hence eco, right? Strain is R, and order in which it was isolated. So this was the first one which was isolated, hence the name 1. Okay, so eco R1. Okay, hint 3 is Haemophilus influenza. Okay, D is the strain name, and 3 is what? You are in the order in which it is isolated. Then BAM H1 is Bacillus amylo. H is the strain, and 1 is the order in which it has been isolated. And these are the recognition sites at which it will be making the cut into the DNA. So they are different. So recognition sequences of restriction enzymes. So the recognition sequence of type 2 endonucleases form palindrome with rotational symmetry. So palindrome with rotational symmetry. So mostly type 2 palindromic sequences. I told you palindromic are those sequences which are mirror images of each other. So if you put a mirror in front of those images, you'll be seeing exactly opposite of the thing, right? So mostly type 2 endonucleases have recognition sites of 4, 5, or 6 base pair, which are predominantly GC rich. Okay? So type 2 restriction enzymes will be cutting at the palindromic sequences, and those sequences would be GC rich. So you see these sequences are mirror images of each other, and that is what they are talking. Then properties of restriction enzymes. So I think we will see about this in the next lecture, because we are about to, we have only 2 minutes left. So are we clear with the idea of restriction enzymes? Do you guys by far know what do you mean by restriction enzyme? What are the types? So these are the concepts that are developed now, and people are working on having more knowledge on this, and how exactly you can make use of this restriction enzyme for good of human beings by doing some molecular changes, molecular level changes. So are we fine till here? Yes, Sumedha, are we fine till here?
Yes, ma'am.
Okay. Okay. Then I will end the lecture here for today, and we let me see from tomorrow if we have holiday or not, because I have not received any notice yet from the college. As soon as I receive it, I will let you know, guys. Until then, we are on the same schedule. Okay, 11:30 and then 1:30. Okay. I'll see you guys, and I'm uploading the videos now on the YouTube. of your presentations as well and my lectures as well. So please have a look at it. And also I'll be updating the material. Okay. Yes. See you all.